Okay, so welcome back to my living room. Today we're going to be covering topic 18, gene expression and regulation. So by the way, this particular topic I have abbreviated quite significantly. If you take a look at the PDF version of my notes, they're a lot longer and a lot more thorough and cover a lot more details than are covered in these PowerPoints. Please do focus on the lecture for your final exam. Focus on what I'm covering here, and I will try to make it clear what is most important for the final exam. So what is gene expression all about? Let me show you this analogy. Here's an exercise. Add the punctuation. A woman without her man is nothing. So I know what you're thinking. All the men in the room are thinking that you're going to add the punctuation something like this. A woman, comma, without her man, comma, is nothing. And all the women are laughing and saying, no way, no way, no. You've got to punctuate it like this. A woman, colon, without her, man is nothing. So if you take a look at this, we have all these words here put together, but you can change their meanings quite significantly by just a couple of commas or a colon or a little bit of punctuation. And gene regulation is much like this. We have 20,000 genes in the human genome, and we have many different cell types. So the way we express our genes can actually make a huge difference in what type of cell you have and exactly how that gene is interacting and responding to its environment. So why regulate genes specifically? There are many reasons. Here are just a few. Like I mentioned in the previous slide, we have to respond to environmental changes. So sometimes it's cold outside or warm outside, so our cells need to be able to respond to all sorts of environmental changes. Sometimes there's a stimulus that we need to respond to. A stimulus could be you're hitting puberty and now you have some hormones that say turn on certain functions. There are many types of stimuli that our body gets. So it says here also to turn on one gene in one tissue and off in another. If you're a blood cell, maybe you want hemoglobin, but you don't want hemoglobin expressed in your skin cells or your neurons. It's important to be expressed in the right tissues and the right cell types. Some genes need to be on more often than others. So our body is constantly making connective tissue like collagen and keratin, and so that these tissues can grow and continue to grow. Other tissues don't need to grow so readily. So let's take a look at a couple of examples of gene expression. I want to talk a little bit about what's going on in prokaryotes and a little bit about what is going on in eukaryotes. If you take a look here, this is something we talked about already, is how we can actually regulate enzymes. We talked about phosphofructokinase when we were talking about glycolysis. Over here, this is an example showing what's going on in the tryptophan synthesis pathway. So tryptophan, you can see there's the word up there. This is an amino acid. And if you take a look right here, we have, it looks like 1, 2, 3, 4, 5 genes, and these genes make enzymes. So this first two genes, they make some proteins. The proteins combine to make a complex enzyme, enzyme 1. Gene number C makes enzyme 2, and the last two genes make a third enzyme. And just like glycolysis, or the Krebs cycle, we have a chemical precursor up here. So here's our chemical precursor, CP, and it gets converted by these enzymes. So enzyme 1 converts it to an intermediate, enzyme 2 converts it from intermediate 1 to intermediate 2, and then enzyme 3 makes our final product, which is tryptophan. And if you remember, with phosphofructokinase, we had end products that were inhibiting an enzyme. So with this particular pathway, we also have a feedback inhibition. So the tryptophan actually goes back and inhibits enzyme number 1. That there is a negative, meaning it is slowing things down. So this is one way to do it. We can have our end products in some sort of feedback inhibition loop that will stop an enzyme or slow down an enzyme from producing more of the actual end product. So this is regulation of the enzyme activity. We're going to come back to this image in a minute. So here's the thing with bacteria, is they basically just have a cytoplasm. So all these processes, transcription and translation, are all happening in the cytoplasm, and they're actually happening quite simultaneously. Take a look at this slide here. What are we looking at? We are looking at the DNA, which is up here. I will draw it for you. Here's the DNA. Right there. And you can see all of these RNA polymerases are coming along with a bunch of ribosomes as well. So what is going on? Transcription and translation are happening at the exact same time. Here's a picture from the textbook. You can see the RNA polymerases here. We have RNA going along right there, and these ribosomes are binding before the RNA is even finished being produced. So if we want to control things in a bacterial cell and we want to do it efficiently, regulating our enzymes is not going to be good enough. It has to be done back at that transcriptional level, back at the before RNA is even being made. So here we are back at this slide here. You can see it's exactly what we showed you before. We have that feedback inhibition. We have tryptophan here, and tryptophan is inhibiting that enzyme. There we go. This is regulation of the enzyme activity. Now we want to talk about regulation of the enzyme production. So we're talking about regulation right at the gene level. So what we're going to do is this tryptophan molecule here, it turns out, can actually regulate production of these gene products. So it's going to interact with the genes directly. This is called genetic control or genetic regulation. We're going to talk about it briefly here today. So let's talk about what happens in bacteria. This is the main thing that happens at the bacterial gene level, are these things called operons. So operon. So what is an operon? You need to know this for the final exam. There will be at least 1 or 2 multiple choice questions on operons. And I will not be asking you about the details of operons, but I will be asking you basically what they are in some form or another. You can see here's a definition that says it's a basic unit of gene expression in bacteria. So how does this work? Here's an operon. You can see what we have here is we have 3 genes. We have A and B and C. And each of these genes are attached together. Notice here this little green thing, this is a start codon. This gene has a stop codon. There's another start codon for gene B, stop codon, start codon for gene C. So all 3 of these genes are basically attached together. One messenger RNA is made. So this here is the messenger RNA. I don't know why it's not labeled. There's my mRNA. And one messenger RNA is being used to make 3 protein products. So you can see the protein product A and B and C. And they're all controlled by one promoter. So hopefully you remember what a promoter is. So a promoter is a DNA sequence. It's found before the gene, and that is where the RNA polymerase binds. So one other thing here, you can see I also have an operator. So an operator is part of the promoter. This is just the regulatory site on the promoter. So it's a regulatory region. So these promoters and operators, these are DNA sequences. It's where the RNA polymerase binds, and its an operator specifically is where the regulation occurs within the bacterial genome. So you can see in my note here, it says it uses one messenger RNA for several genes. So we're going to multitask. We're going to control several genes all together at once. Here's the definition of the operator. It says a segment of DNA. I already mentioned that. This is within the promoter, and this is where a regulatory protein, which is called a repressor, can bind, and the repressor will be able to block the RNA polymerase. Let's take a look at a picture. Here's a picture I found on the internet. You can see there's the RNA polymerase it's trying to bind. Here's the RNA polymerase it's trying to bind, and here's the repressor, and the repressor is blocking the RNA polymerase. Here's a definition of the repressor, a regulatory protein that blocks the RNA polymerase. Let's take a look at a couple of examples. So here is the tryptophan synthesis operon. Normally I go through this in class in quite a bit of detail. I will not be testing you directly on the tryptophan synthesis operon, but I'm showing it to you right now as a good example. So notice we have several things here. The operon is labeled here. Tryptophan operon right there. Take a look at what we have. We have a promoter and operator, and there's our RNA polymerase. It's binding, and it's going to make the RNA for all of these genes. Secondly, notice each of these genes, they all have start codons and stop codons, and then there's the protein products. So each of these genes, you can see there's 5. I don't know why they're labeled A, B, C, D, and E in that order, or E, B, C, B, and A, but that is not me who named them. Notice over here on the left, we have another gene. That's the regulatory gene. It makes this regulatory molecule here called a repressor, which, like I said, I'm not going to cover in really a lot of detail. I'm just trying to show you that all these things are regulated at once. So, like I said, it's a way to multitask so that many genes can be turned on or turned off at once. So in this case, all these genes have a common goal, which is to make tryptophan, so we turn them on or off all at the same time. There's my note. Will not be on the final exam. The tryptophan synthesis operon will not be on the final exam, but operons in general will be. So here's another example of an operon. This is the lactose operon, or the lac operon. If you take more genetics classes, you will talk about this in lots and lots of detail, believe me. I just want to show you it as a second example of an operon. Notice it has 3 genes. We've got 1, 2, 3, lac Z, lac Y, and lac A. We've got 1 messenger RNA, and we've got 3 proteins. So the production of 3 gene products are controlled all by one operon. There's our RNA polymerase right here. It's binding the promoter and the operator, which are found right here. We also have this repressor protein, which is activated in the presence of lactose. I won't be asking you about that. So just a reminder, this specific operon will not be on the exam, but I will ask you about operons in a general sense. So make sure you know a definition and kind of generally what is going on here, that you have these multiple genes being regulated simultaneously. So let's talk about eukaryotic cells for a bit now. Things in eukaryotic cells are, of course, a lot more complex. So we have our transcription that happens in the nucleus, and of course we have all sorts of RNA processing that happens in the nucleus. So remember, we have introns that are cut out, we have the capping and tailing, among other things. Eventually, the messenger RNA is going to be exported out a nuclear pore, and then in the cytoplasm we have our translation at the ribosome. Of course, the last thing to talk about is how we get those proteins to specific organelles. So this will be something that will be very important that we will be talking about in this lecture. So if you think about all the processes that happen in eukaryotes, we have our DNA, it becomes RNA, it gets processed, becomes messenger RNA, the messenger RNA finds a ribosome, becomes a protein, the protein isn't necessarily active right away. All of these things, at each of these stages, there are opportunities for gene products to be controlled and regulated. All of these have various things that are covered in the textbook. We're not going to be looking at most of these things in any amount of detail. Mostly I will be discussing things that we had talked about already in previous lectures, and I will be talking about translational control. So make sure you take a look at this part of the notes. We're going to be talking about something called the signal peptide and how things can be sent to the endoplasmic reticulum to those bound ribosomes. So here's one thing that we did talk about already. Remember, we talked about the U chromatin and the heterochromatin. Remember, U means true. So this is the chromatin that is a little bit more diffuse, and hetero means the condensed chromatin. So let's see if we can find some of these things. So what are we looking for? The nucleus? So obviously this here is going to be the nucleus. It's this nice big thing here. U chromatin. So the U chromatin is this stuff here. Here's an E. And the heterochromatin, let me try a different color, is this stuff right here. There we go. What else are we looking for? The mitochondria. So there's a mitochondria here. Maybe I'll try a different color. Thought I saw there's another mitochondria down here. Red looks pretty good. Rough endoplasmic reticulum. So up here we can see the rough ER up there. And the Golgi complex. A little hard to find, but it is up here. You can see those little blobby vesicles kind of coming off. Here's somebody else who labeled it for me a little bit more professionally. So what's the big deal about the euchromatin and the heterochromatin? So it turns out the heterochromatin is tightly packed and it's very hard to get at those genes. So some genes in some cells are turned off just simply because they're packed away and they're not accessible. So some chromosomes in some cells are turned on and activated, and some chromosomes are turned off or deactivated, or at least parts of those chromosomes can be deactivated in some cells. Something else we already talked about as well are these transcription factors. So remember way back we were talking about transcription, and we were talking about the differences between prokaryote and eukaryote transcription. So these are found in eukaryotes. So what are transcription factors? Transcription factors are these things that are required for transcription in eukaryotes, partly because we are much more complex. We have different tissues, we have different developmental stages, and so transcription factors are essential for the RNA polymerase to be able to start. So notice here, general transcription factors, it's helping the RNA to bind. There's that TATA box, there's RNA polymerase, and this whole thing is called a transcription initiation complex. There we go. There's the note. The transcription initiation complex is composed of general and cell or tissue-specific transcription factors plus RNA polymerase. There we go. RNA pol, we'll call it for short form. So here's a model from the textbook. It's quite complex. Notice there are a lot of things here that I'm not talking about. There's enhancers and activators and silencers and all sorts of other things that we're not going to cover this particular semester, but I am going to point out the things we have talked about. So there's the gene over here in blue, and before the gene we have a promoter. Of course, promoters are always there before genes. That's where the RNA polymerase binds, and we have our TATA box. So the next step, notice we have some general transcription factors. There they are. Give them a little wiggle. And we have a bunch of other proteins that are helping the RNA polymerase. I'm not going to talk about them really at all. There comes RNA polymerase. There it is. And then lastly, the RNA polymerase binds along with those transcription factors, and this is the transcription initiation complex. And then the transcription, of course, is going to go in that direction along the gene in the 5' to 3' direction. Okay, so what I do want to talk about in the most amount of detail here is this one here. So notice I put a little star beside this, targeting to specific cellular locations. There are lots of ways that proteins can be modified after translation, after they're made. You can see I have a list of some of them. I'm going to talk about a couple here and there. Some are cut, so proteolysis. Some are glycosylated, meaning adding carbohydrates. Some we add phosphate groups to, phosphorylation. Some become lipidated. They become glycolipids. So I want to talk about a couple of these, but more detail I want to talk about this one here. So one thing that many proteins need to do is they need to assemble before they become fully functional. So remember ATP synthase found in mitochondria and chloroplasts? So there it is. It has about 15 different subunits, so all 15 of those subunits have to be assembled together before you have a fully functional machine. So that's something important for a lot of proteins. They need to be assembled first. As I mentioned, some need glycosylation. So if you take a look what we're looking at here, these are the blood type antigens. So we have the O antigen, the A antigen, and the B antigen. So if you have A type blood, you have this carbohydrate here. So this is N-acetyl galactose. If you have the B blood type, you just have galactose, not that N-acetyl group. If you have the O blood, you don't have that antigen at all. If you have AB blood, you have A and B antigens on your red blood cells. So remember where glycosylation happens. It happens in the ER. It starts there and it finishes off in the Golgi body. So if I ask you on the final exam, where does glycosylation happen, you can answer out of those, endoplasmic reticulum or Golgi body. Okay, so let's talk about this targeting to specific cellular locations. So remember we were talking about ribosomes, and we have the free ribosomes. These are for cytoplasmic proteins, So these are found in the cytoplasm. They're not bound to anything. And then there's the bound ribosomes. These ones are attached to the ER, and these are making proteins that are being sent to any part of the endomembrane system. So they can be sent back to the ER, they can be sent to the Golgi, lysosome, vacuoles, sometimes the nucleus, mostly the nuclear membrane, and anything that is secreted or exported out of the cell. So here's an example of a protein you might be familiar with. This is insulin. Insulin is produced by your pancreatic beta cells, and insulin is made in the rough ER and then secreted from the cell. So something any of these cells have is this thing called a signal peptide. So there's a definition we're going to have to take a look at before we can understand exactly how this works. So what is a signal peptide? So signal peptide is an amino acid sequence, and it's found at the beginning of a newly synthesized protein. So the beginning is called the N-terminus. You can see that terminology I have here in the PowerPoint. N-terminus, N is for nitrogen, which is found in the amino group at the beginning of an amino acid sequence. So it's a short amino acid sequence present at the beginning of a newly synthesized protein, and specifically these are proteins that are destined towards secretion or the endomembrane system. So here's a picture of one showing a signal peptide. It has a few things. It has some positively charged amino acids. It has some hydrophobic amino acids, so usually about 15 is typical. A few uncharged amino acids. There's a cleavage site. So this particular thing, the signal peptide, is kind of a little flag, and it says, hey, come grab me and bring me over to endoplasmic reticulum. So that's really what it is. It's a signal. It's a flag, it's a marker, whatever you want to call it, and there's actually several names for signal peptides, but this is the terminology used in our textbook, so I'm not going to deviate from that. But like I said, it's a signal that says, hey, come grab this protein, bring it over to the ER. You can see the last part of this is we have the actual protein is coded after the signal peptide. This is the actual part of the protein that is going to carry out its biological function. So let's take a look at this in more detail. So this is the image from the textbook. It's actually quite complex, so I tried to break it down into several steps so that we can talk about what is going on here specifically. Notice we have a ribosome there, and on that ribosome we have some messenger RNA. And so this is actually where all ribosomes start. All ribosomes start in the cytoplasm. They're not naturally bound to the ER. So notice here we have our signal peptide. Remember what that is. It's just a few amino acids, so about 20-ish amino acids. And they say, hey, come bring me over to the ER. So what is going to bring it over to the ER? Well, this little orange thing here, this is the SRP. So what is the SRP? The SRP stands for Signal Recognition Particle. So you can see that the name fits. Signal recognition. So it's recognizing the signal peptide. It's attaching to it. It's going to bind to it. What's it going to do? So it's a protein RNA complex. That's why we call this a particle. SRP does not stand for signal recognition protein. It stands for signal recognition particle because it's not just protein. It's actually made out of RNA. We'll talk about that RNA in a few minutes. That's the last type of RNA we need to talk about in this class. So what does it do? It binds the signal peptide. We mentioned that already. And then lastly, it brings it over to the endoplasmic reticulum. So here's the ER here, this blue thing. There's the ER membrane. Just draw a little line here. The inside of the ER is called the lumen, by the way, if you didn't know that. And the outside is the cytoplasm. So here goes. The SRP is going to bring it over here to this SRP receptor. So there's a little receptor on the ER membrane. And here we go. It brings it over. So there is the SRP taking it over to the ER. So look at my note there. It says the signal peptide is found at the N-terminus of a protein, so you know what that means now, and it is bound by the signal recognition protein. So let's go to the next step. So let's take a look at what's happening in this next step here. Here's my note. It says, once the signal recognition particle docks on the ER membrane, translation continues. So something I did not mention before is that when the signal recognition particle actually binds the signal peptide, translation stops. it's put on pause for a bit until this whole thing can dock with the endoplasmic reticulum membrane. So here goes. Step 3, you can see it docking with that receptor. And this receptor actually has basically some sort of pore in it. And when translation resumes, so we'll go to step 4, you can see when translation resumes that the protein is actually being made inside the ER. So there it goes. That's supposed to be an arrow. Step 5, notice that the signal peptide is removed. That was that cleavage site. We don't need the signal peptide anymore. We've already brought it over to the ER. The protein continues to get translated. There it goes. And here we go with the last step. So now we have our protein. Notice the protein is made inside the ER, and our ribosome, it departs. It's going to get recycled along with the messenger RNA. So take a look at that. I told you it was quite complex. Make sure you know these definitions. Make sure you know what the SRP is. Make sure you know what the signal peptide is, and have a basic idea of how this works. You can expect either a multiple choice or 2 or a short answer or 2 on the final exam for this. So I just want to take a look at the last thing, why we are inside the ER and how that works to getting a protein exported or secreted outside of a cell. I did not have a good diagram for this. Usually I draw something on the board. I was able to find something on the internet that I was able to modify to try to explain what is going on here. So we're starting right at the beginning. So we've got our messenger RNA, right? It exits the nuclear pore. There it goes. What happens next? It binds a ribosome, and then, of course, if it has that signal peptide, it is recognized by the SRP, which is going to take it over to the rough ER. So in the next step, you can see this is basically what we talked about in the previous slides. We've got, here's our little ribosome right here, and it's found on the outside of the rough ER. Our protein is being produced. There it goes. It's being made. It's inside the rough ER. So it's that little green blob right here that I'm just trying to draw an arrow to. So it's inside the rough ER. So at this point, now you can see our protein, there it is, it's being put into one of those membrane vesicles. So it's going to butt off from the ER, and then there it goes. It's going to go down. It goes to, of course, the cis side of the Golgi is the part that is near the ER. It travels through the Golgi. This process isn't fully understood how it travels through the Golgi, but eventually it's going to emerge on the trans face of the Golgi. So there it goes. We have another membrane vesicle, and this membrane vesicle here, it buds with the plasma membrane, and our protein in the end is secreted or exported. So there it goes. Okay, this almost wraps up our topic. Just a couple of other things to mention. Like I said, it's really, I must emphasize that control of gene expression can be done at many, many different stages. You can start right at the DNA level. A big part of it happens at the transcriptional level. Some things happen at the translational level. Some things are regulated at the ribosome. Of course, there's splicing and capping and tailing. There are many areas where gene expression can be regulated. Okay, so we were talking about those types of RNA in the last few units. I told you there would be a question on the final exam addressing the 6 types of RNA. So take a look at this slide. We have our messenger RNA. This carries information from the DNA to the ribosomes. We have our transfer RNA. This carries the appropriate amino acid to the ribosome during translation. We have ribosomal RNA. This deals with structural and catalytic roles in the ribosomes. With telomerase RNA, this is used by the telomerase enzymes as a template to extend the ends of linear chromosomes. We have small nuclear RNA. We talked about these with regards to splicing and spliceosomes, structural and catalytic roles again. We also have our SRP RNA. This is the new one. So remember what SRP stands for. That's signal recognition particle. So what is this? This is a component of the SRP, a component of the signal recognition particle, and it helps to recognize the signal peptide, and then carries out that function of bringing that particular protein during translation over to the ER. So make sure you know all of these types of RNA. You know their names, you know their definitions, because I'm going to ask you about this on the final exam. So make sure you know this. This will be 6 smart questions, so know this for sure 100%. So that finishes topic 18. Like I said, take a look at the notes and then take a look at these PowerPoints, because I have cut out quite a bit from the notes, and I want you to really focus on what I've talked about in this particular PowerPoint. There's a few more things for you to learn. Don't worry about these cytoplasmic determinants. We did not talk about those. Let me just take a look at these definitions. So we did talk about operons and operators. We previously talked about promoters, the tryptophan operon and the lac operon. Like I said, we aren't going to be covering information about those directly. We can cross off enhancers, polyribosomes, repressors. We did talk about signal peptides, SRP, not a cytoplasmic determinant. We did talk about different types of ribosomes and kind of talked about regulatory proteins. Here are some study questions for you. Make sure you take a look at them and take a look at these details here. We just did not really talk about differentiation, but we did talk about everything else. So this is the end of lecture on topic 18. We have 2 more lectures to go, and the last 2 I will also significantly abbreviate and make quite a bit shorter, because I really just want to focus on the essential information so that you guys are set up for second year. Have a great day. I hope everyone is doing well.